Before I introduce Howard, who will take the rest of the evening here, I just want to comment a minute about the ADDF and what you're going to hear tonight. Leonard Lauder, over a decade ago, came up with a phrase when he was talking about the ADDF. He called it, "Hope is on the horizon." And tonight, you're going to hear that hope is actually here. And I know that's something you don't hear a lot about, hope with Alzheimer's. I'm just going to give you the 30-second elevator spiel about the ADDF. Founded 24 years ago by the Lauder family, they made a decision. They were only going to fund science and a certain type of science. It's called translational science. How do you go from the bench to the bedside? How can you speed interesting science so that it's actually treatments and preventions and ultimately cures? And the Lauder family decided that they're only going to fund what often is called valley of death science. Interesting ideas in the lab, but they're not so de-risked that big pharma is coming in to take them through to patients, right? So somebody has to pick that mantle up. The Lauder family did that about 25 years ago. The Lauders also decided they would not give out grants. Rather, they would give out money to academics and to biotech companies in the form of investments. And those would go as investments, so if that academic institution or that biotech saw some kind of commercialization of what was being funded, the ADDF would get a part of that and then put it right back into the science, so a way to speed the science in a very innovative way. And the other thing that I will let you know is that the science you're going to hear about tonight is called, a big banner over it, the biology of aging. And what do I mean by that? I am delighted to see that I'm staring at two engaged men. One in the first row, Larry Leeds. Larry, put your hand up again. The other in the second row, which is Josh Lauder, put your hand up again. And you may notice there's a difference in their ages. But I will assure you, while we sit here, both of them are aging. We are all aging. It's true. And we age in different ways, okay? And Dr. Fillit, who you're going to hear about, talks about 7 different pathways that are involved with your aging. Could be genetics, could be something like inflammation, could be vascular, could be any number of things. And what the ADDF for the last 10 plus years has been funding this concept that we all age, but if we age and our memory starts going, if we age and we start forgetting our children's names, if we age and we can no longer communicate with the outside world, that's not normal aging. And so what Dr. Fillit and the board of the ADDF came up with, this biology of aging, and we're going to fund all these shots on goal across these 7 areas, and we're going to see where it's going to take us. And the last thing I'm going to say before I turn it over to Dr. Fillit is the following. I know today, in my lifetime, right now, if my cognition started to go, I have options. I have 2 children who are both married, late 20s, early 30s, and I know in their lifetime, they're going to have prescriptions around prevention, prescriptions around combined therapies, and something called precision medicine. And I'm also thrilled to tell you that I have a 9.5-month-old grandson. He's the cutest baby ever. And I know in his lifetime, so long as he stays on prescriptions that doctors would give to him if his cognition started to go in his 60s, 70s, 80s, he's not going to die from Alzheimer's. Not a lie here. This is exactly what the ADDF is funding today. Last piece, thank you to the Lauders. Because of the Lauder family, today, yesterday, tomorrow, anybody who donates to the ADDF, the Lauders take care of all of the expenses, so every nickel of the donation goes directly to the science you're going to hear about tonight. It's enough of me. I want to give you our co-founder with the Lauder family of the ADDF, Dr. Howard Fillit, who's given his life to the science of aging, his life to the improvement of cognition. He's our co-founder and chief science officer, Dr. Howard Fillit.
Thank you, Mark. Thanks, everybody. I thought I'd give a little bit of an overview of where we've come in just in my professional lifetime. I saw my first patient with Alzheimer's probably in 1979, which was about 40-something years ago. Up until that point, I had been training in medical school, seeing patients in an internship, a residency, fellowship, taking care of hundreds and hundreds of older people, and never made the diagnosis of Alzheimer's. In fact, I never heard of it. And when I looked back in my textbooks, Alzheimer's disease was not in my medical school textbooks. And the first time I saw a paper with Alzheimer's disease was when I had the privilege of visiting what's called a teaching nursing home. I was interested at that time in geriatric medicine, and I walked into this very beautiful architecturally designed building that had 300 rooms, 300 beds in it, and they were all very nicely with furniture and all that. And they took me on a tour, and I saw these people that had Alzheimer's disease, and they were bedridden and couldn't walk and couldn't talk and couldn't bathe themselves. And for me, I actually wrote and published my first neuroscience paper in 1971, which is almost 50 years ago. And so I pursued neuroscience research. And putting those two things together, a love of older people, geriatric medicine, discovering this disease of Alzheimer's, and a love of neuroscience, it just became natural for me to kind of spend my life at. So when I started, nothing was known about Alzheimer's disease. I mean, literally, we knew nothing about Alzheimer's disease. We knew what Alzheimer had described in 1906, but what happened was that Alzheimer described dementia in a 52-year-old woman who was clearly, because she had dementia, she was obviously had a disease, but up until about 1968, 1970, senility was thought to be a normal part of aging. So in other words, when people started losing their minds and becoming childlike, everyone thought that this was normal. And what changed was that some pathologists working in London in the late 60s decided to do a very simple experiment. They looked at the brains at autopsy of older people who died with dementia, with senility, and they looked at the brains of older people who died without senility. And what they found was the same pathology that Alzheimer described in the 52-year-old woman, the plaques and tangles, in the old people who were senile. And so it was on that day in human history that senility went from being thought of as a normal part of aging, oh, grandpa's senile, to being understood for the first time as a disease of old age. And when my mentor, Robert Butler, realized this, he wrote a book called Aging in America and got the Pulitzer Prize in 1976 for writing that book, and then became the first director of the National Institute on Aging. And when he went there, he looked at how much money was going into Alzheimer's research in 1976, at a time when we were spending as a nation billions of dollars on the war on heart disease and the war on cancer, and we spent $625,000 on Alzheimer's research. And I say these historical things because Alzheimer's research started really in the early 1980s, which was, I guess, about 40 years ago. That's not a long time in the history of research for a disease. The beta amyloid protein that everyone knows about, plaques and tangles and all the proteins today that are in those plaques and tangles, that wasn't discovered until 1984. And the gene discovered in 1986. And around the same time, a molecule called tau was discovered in the dying neurons in the tangles. Today, just this past year, in the last few months, you heard about Biogen's drug and how there's a lot of controversy about how the drug came about to get an FDA approval. But the fact is the drug worked. And it's really a breakthrough. And I just want to tell a quick story before I turn this over to Frank. And it's kind of a story of not only what happened in my professional career, but what the foundation was able to do, a small foundation at the time, to make a huge contribution to the field. So in 1999, somebody called me up from the University of Pennsylvania and said, I have a crazy idea and I can't get any money for it. And I said, what's that? And he said, I want to take the same dye that Alzheimer used in 1906 to stain the brain of that 52-year-old woman and discover Alzheimer's disease, the plaques and tangles. And I want to put a radio label on it, and I want to be able to inject it into the artery of a patient. It'll go up into the brain, it'll bind to the plaques if it's there, and it'll show up on a simple PET scan. And the beauty of it is that we won't have to do an autopsy to make a definitive diagnosis of Alzheimer's disease. We'll be able to do it with a brain scan. And so ADDF started funding that program from 2000 to 2004 at the University of Pennsylvania. In 2005, the program was spun out into a small biotech company called Avid Radiopharmaceuticals with about $7 million in venture capital. In 2010, that company was acquired by Eli Lilly for $800 million in milestone payments, of which we got royalties on. And in 2012, that test became the first test approved by the FDA for the diagnosis of Alzheimer's disease. And today, if somebody comes into my office with memory problems and they want to know definitively if they have Alzheimer's disease, I can send them down the block to the radiology office, and by the next day I can tell them whether or not they have Alzheimer's disease. Now that really has changed to some degree clinical care. It's expensive and it's not paid for by Medicare. But what it really did is, if you heard about this recent drug from Biogen, the drug got what's called an accelerated approval, which means that the drug had a pharmacology to remove the amyloid from the brain and test the theory that if you could remove the amyloid from the brain, you'd slow down the disease. Now, how are scientists going to be able to see if you remove the amyloid from the brain? They used our test. Everyone who went into that clinical trial for the first time had to have a positive scan. That was a big advance. And then the people were administered the drug, and the test was used to see if the drug was working. Well, the drug worked. And 80% of people that got the drug at 18 months, their brains was completely cleared of amyloid. We could tell from the brain scan. And not only that, but their tau biomarkers got better. So we know that that drug works because of the biomarkers. And there's a signal there that it looks like the disease is slowed down. This is, in my opinion, although the drug might not be clinically useful at this point, the drug got an approval based on the biomarker test. There is also a blood test on the market now in 49 states, not in New York, but in 49 states. You can go to your doctor and get a blood test and find out if you have Alzheimer's disease. We've invested in that for over 10 years. I tell you this because, in conclusion, let me just say that we're funding over 30 clinical trials in different areas of, as Mark mentioned, inflammation and so on. But what's exciting is that we haven't had a drug approval in over 20 years in Alzheimer's space. We've had failure after failure after failure. But the field has matured from nothing, I mean literally nothing, 40, 45 years ago, to where we're in a really exciting time. We have biomarkers. We can see if a drug is working. We can subset patients according to their profiles. And we can test drugs. There's over 100 drugs in clinical trials. And many of these are not the amyloid drugs that you're hearing about from Biogen and Lilly and Roche. These are in all different areas of research. As Mark mentioned, aging is the leading risk factor for Alzheimer's disease. And there are these various pathways that the cell goes through. That's why cancer is a disease of aging, and people with cancer need to be on 4 or 5 drugs to attack all the pathways to conquer the cancer. People with diabetes are on 3, 4 drugs. People with hypertension. We're going to have the same thing with Alzheimer's, but we've made that first step. I really believe we have the first step. Now, the thing I'm most excited about, and we've been doing this for over 20 years, is working with Frank Longo. And I'll tell you why. Because brain cells can get injured many different ways. They're being bombarded every day. And through evolution, brain cells and other cells in our body have developed protective mechanisms to survive and fight all these different ways the brain can be injured. Well, there are hormones in the brain. One's called NGF, and the other's BDNF, and Frank's going to tell us about that, which kind of say to the cell, regardless of the form of injury, we're going to activate these signals that protect the cell from dying, or if the damage is so bad, the cells go in and basically commit suicide to get out of the way. And I'll let Frank tell you why this is so exciting. But this drug could really be revolutionary. And we started working with Frank on this back in the year 2000. Frank is the chairman of Neurology and Neuroscience at Stanford University Medical Center. Prior to that, he was chairman of Neurology at University of North Carolina in Chapel Hill. And before that, he was at UCSF. And we've been working with him all through that period of the last 20 years and helped him start a biotech company. And it's just been a fascinating journey to work with Frank. So, Frank, maybe you could start us off by telling us how the project got started and what it's like to develop a drug. And also, I want to say that Frank, being chairman of Neurology and Neuroscience, the co-founder of a biotech company, and a leader in drug development, he's a classic physician-scientist. I mean, we all really respect Frank for what he's doing and brings the whole 9 yards to this effort.
Well, thank you, Howard. And I also wanted to add my welcome to everybody. Thank you for being here. It's been a pleasure to work with Howard and his team and the ADDF over the years, and appreciate the Lauder family support and all the other supporters here. So thank you. So Howard, your question, how we got started, I've been interested in the brain for a long time, in the biology of the brain. I've been interested in regeneration. When I was younger, I was told that there could be no treatment for a brain injury or brain disease. And so once I was told that, then I set that as my life goal, is to make therapies for the brain. And right before I met Howard, we had noticed in Alzheimer's disease that it seemed like there were mechanisms in Alzheimer's where the brain started degenerating, losing the connections between neurons that were similar to how the brain's built in the first place and how the brain's wired in the first place. And so we had an idea of creating therapies that would harness those very powerful mechanisms to protect the brain. And I can share more details with that, but to make a long story short, I applied to NIH for a grant to pursue this. And NIH has to be fairly conservative about what they'll fund. And at this time, about 20 years ago, there was a lot of excitement emerging about amyloid. As Howard mentioned, the amyloid accumulates in the brain in Alzheimer's, and people were starting to come up with therapies that might be able to lower the amyloid. So that's what was exciting. So when I applied to NIH for a therapy approach that did not involve lowering amyloid, they thought it was pretty interesting, but not quite in the fundable range. And so next thing I knew, I got one of the best phone calls I ever got from Howard Fillit. He can tell you about that, but introducing me to ADDF and a possible way to get this new area funded.
So Frank was the way you start out is you try to get molecules, right? That's how you develop a drug. I mean, it's so complicated, it's unbelievable. Imagine you're trying to develop a little white pill that's going to cure Alzheimer's. I mean, and 90% of people that work in the pharmaceutical industry spend their entire careers never working on a drug that comes to market. 90%. Because it's so hard and failure is the norm. But people love science. We love the process of science. And what Frank was doing to develop his drug was the very beginning. So he bought a computer library. Maybe you could tell us about that.
I can tell you about how we got started. So there are naturally occurring proteins in the brain that dictate whether a connection between the neurons will be maintained. So when our brain develops, we actually have more nerve cells and more connections that we need, 2 or 3 times more. And so then there's what's called a pruning process, like pruning a tree, where we get rid of many of these connections so the brain works better. And so there are mechanisms inside the nerve cells that really carefully control whether a connection between the nerve cells will persist or not. And there are some naturally occurring proteins that are very powerful regulators of this process. Howard mentioned them. One's called nerve growth factor, or NGF, and the other one's called brain-derived neurotrophic factor, or BDNF. These are 2 very powerful proteins that dictate keeping a synapse or not. And in fact, the discovery of these powerful proteins was done by a scientist named Rita Levi-Montalcini, an Italian scientist, and she won the Nobel Prize in 1986 for the discovery of these very powerful brain proteins. And when she gave her Nobel Prize acceptance speech, one of her main remarks that I noticed was, well, it's great that we discovered these proteins. I hope someday in the future somebody will figure out how to make a drug out of these proteins. Because a protein itself can't be a drug for the brain. It's too big to get through the blood-brain barrier, so it can't be used for a drug itself. So what we wanted to do is create drug-like compounds, or small molecules we call them, that could be just as powerful as these naturally occurring proteins and do what they do to tell the neurons to maintain their synaptic connection and to protect those neurons against whatever might come along, whether it's amyloid or tau or inflammation. We thought these drugs could protect the neurons against those things. So we set out to find and make drugs that do what these proteins do. And the old-fashioned way of doing that is you could test physically thousands and thousands of compounds to see if they would have the activity that these naturally proteins do. And this could take 10 or 20 or 30 years. This could go on forever, and nobody would want to fund that. So I brought in a friend of mine that's a software and computer genius, much smarter than I am, Steve Massa. And he and I figured out how to use this software to screen drug libraries on the computer for hundreds of thousands of compounds every day. And we ended up finding these small molecule compounds, drug-type compounds, that had the exact same shape of the critical part of these powerful proteins. And so we were able to obtain these. We hired medicinal chemists to work on them further, and we discovered that we found a few that had very powerful ability to protect neurons from dying. And then the next step was to move into the mice. So it's a long process, but that's how we got started.
Right. So what took a year of funding from us, you say can be done in about a week now, I think you told me.
Yeah, the good news is it was what we spent a lot of time doing. All these technologies have improved, and this can be done much faster now, more efficiently.
Right. So we went from the chemistry, you screened about 2 million compounds, as I remember, and out of the 2 million, you found about 60 that looked pretty good. You bought the powders from a company that sells the chemicals, and then you tested them in test tubes to see if the powders of those chemicals protected cells from amyloid injury. Right. And then you found 2 out of the 60 that protected well enough that you went into mice that had Alzheimer's engineered into them.
Right.
And so now we're going from like the year 2000 to probably 2010, 2015, something
That's right,
like that.
yeah.
But along the way, while you were at UNC, you and I were talking, and this was really exciting. And I said to you, Frank, why don't we start a biotech company? So you did. And
Yeah. So at that point, we had one and several backup compounds working in Alzheimer's mice, and that was really exciting. And you could build your whole career on just that. You publish a paper saying the compounds work in mice and that these could be a treatment for Alzheimer's someday, and you get promoted to full professor, and that's it. But we were determined to get these into people, and now now the hard work really starts. How do you go from a mouse to a person? And I could see that it would be very difficult to do this in a university laboratory environment. I mean, the university laboratory is great for a lot of things, but moving something into a commercial or clinical part is much more difficult. And we were too early to hand this over to a pharma company at this point. It would have been too risky for our friends in pharma. So the only option left was start your own company, license it to your own company. And of course, I had no idea how to start a company. And that's where Howard and the ADDF were very encouraging. Frank, if you start a company, we can fund it. So, wow, okay. So I found an attorney, and in one day, the initial name of our company was Longo Pharmaceuticals. But then the university said they wouldn't license the technology to Longo Pharmaceuticals unless we had funding. And so our first funding came from ADDF, and then the whole thing could get started that way.
so we were first investors, and we've continued to invest along the way, and it's now almost 15 years later, and the compounds worked in mice, which is pretty amazing. And then you got into the regulatory science, which is a whole nother thing, like what you have to do to take a molecule and be able to give it to human beings with all the FDA regulations, prove its safety and then go into phase 1. And that was a whole nother thing.
Yeah, moving from mice to people involves a lot of safety, which is good. So the FDA requires that a new drug is first undergoes additional animal testing in rats and other animals. I mean, it's all, I think, done very respectfully to the animals. I learned a lot about it. And fortunately, all of that, and this took 2 or 3 years, all of that animal testing was very safe. The animals did well in our drug, and they're monitored very carefully. And I learned about how important this is, how challenging it is. And for an academic person, it's not exciting. You don't publish a famous paper because your drug was safe in a rat. But yet it's so critical before we move to people, right? So all that went well. And then it was time to meet with the FDA to see if we could get permission to start our first human study. Now, because our drug was entirely a new category, new class of drug, and the target we were going after in the brain with this drug, nobody had ever gone after before, the FDA required that our very first trials would have to be in so-called normal people, not Alzheimer's patients, but normal people. So that was a new area. Then we had to get funded what's called a phase 1 trial, phase 1 in normal people, just for safety. So that was the next step that I can tell you about. But there are different chapters along the story here.
Yeah. Well, the most exciting thing is that after proving that the drug was safe, for the first time, and we're talking like 15, 20 years later, and by the way, during this time, Frank's a practicing doctor. Frank's chairman of neurology and neuroscience at Stanford. I mean, I don't know how you do it, but...
Yeah, not much sleep. So the very first time our lead drug, we're developing many drugs, was given to a person was a very emotional experience for me. How this is done, there are companies that help with the first test in humans. And so this particular company was a well-established company in Kansas called PRA. And so people volunteer to be first to take a drug. And so we had 72 normal volunteers in our phase 1 trial. Younger people and older people, all fairly, none of us are perfectly normal, but all fairly normal people. Nobody had Alzheimer's in this group at that point. And so the volunteers come in, they stay for 10 days in an inpatient type of setting where they're monitored 24/7 by doctors and nurses. And our drug, fortunately, is a capsule. You take it by mouth. It's not an intravenous, it's a capsule. So so I got to be there. I wasn't directly involved, but I got to be there and watch everything. So the very first person to take our drug was a gentleman in his 20s, young guy. And when you first take the drug, they put it in a paper cup. They give you a glass of water. You've got nurses and doctors there. You've got an EKG on. Your blood pressure's being monitored. They're going to be taking your blood. They're monitoring everything. No human being has ever taken this drug before. And of course, for me, I'm a physician. The first rule, do no harm. At this point, I just didn't want anyone to get hurt taking this very new drug that we had invented. And so he put the capsule in his mouth and he took the glass of water and swallowed it. And believe it or not, they made him open his mouth, and with a flashlight, they look and see if he swallowed it. And so he did. And then they started measuring his blood pressure and everything. And fortunately, everything was fine. Blood pressure, the vital signs, the blood tests were all okay. And they were for everybody else. And afterwards, I got a chance to talk to him. And I said, what made you volunteer to take this drug? You don't even have any medical problems. This could be dangerous. I mean, usually it's not if it's safe in rats and dogs. And he said to me, Dr. Longo, my grandfather raised me, and he died with Alzheimer's. And it's the least I could do. And it just really shook me up what that meant. So this is a team effort from him and all of us as well.
So Frank, you recently did the phase 2 trial in a few hundred people, I think, in Europe, and reported that results of that trial of this, what I think is a revolutionary drug, which would be good not just for Alzheimer's, but other neurodegenerative diseases, potentially Parkinson's and others. And you reported on your results in Boston, I guess a couple months ago. where we actually looked at whether the drug works. And that's probably the most exciting thing that you're going to tell us about, because I think you did get some positive results, but we still have challenges. So tell us about that.
Yeah, so I described the phase 1 trial for safety in normal people. Phase 2 is the first time in Alzheimer's patients at a smaller scale, and then the phase 3 would hopefully be the last trial. So Howard's described it. We just finished phase 2 in Alzheimer's patients at the mild to moderate stage. So we decided to do a trial with people in the mild to moderate, somewhat more advanced stage than what many of the trials are. Many of the Alzheimer trials nowadays are much earlier stages, as you probably know. But what we were finding in our Alzheimer's mice is that we treated some of the mice at late stages on purpose. Why did we do that? We really think this drug has a very strong biologic effect. And what we were seeing in the late stage Alzheimer's mice is some reversal of the loss of synaptic connections. So you'll always hear that, oh, we have to treat early, this can only be prevented, that's the only solution. I don't agree with that. I think if the biology of the drug is powerful enough, what we're seeing in the mice, there's some reversal component too. So we wanted to go to later stage in people to really find out how powerful, can this drug do something in a later stage person? Because if it can do something there, it's going to be even better in early stage. So mild to moderate, later stage, and 241 subjects, 1/3 of them on placebo, 1/3 low dose, and 1/3 high dose, blinded trial. Nobody knew what they were on, or the neurologist evaluating them didn't know. We treated them for 6 months. And that's a limited time frame, but that's as far as we could go in a phase 2 trial the first time you're in the disease. Now for phase 3, we'll treat for 18 months, a longer period. And our question was, we had 2 questions. Was it safe in Alzheimer's people? And number 2, did we see any evidence for slowing down the progression that we would expect to occur even over a 6-month period? Unfortunately, for most people, mild to moderate Alzheimer's, even over 6 months there's some progression. And we measured the progression using 3 techniques. An MRI brain scan, looking for parts of the brain that shrink over those 6 months. What we call an FDG PET scan that measures metabolic function in the brain and measures how well those synaptic connections are communicating with each other. And then we did a spinal tap looking in the spinal fluid for proteins that go up with degeneration. So the more degeneration there is, the more these proteins go up as the neurons are degenerating. In all 3 of those areas, MRI brain scan, FDG PET scan of the brain, and then the spinal fluid proteins. In all 3 areas, we found evidence that our drug was significantly slowing down the degeneration during that 6 months compared to the patients that were on the placebo. And this is really exciting. One of the top people in the world that measures those spinal fluid proteins, his name's Kai Blenau, he's in Sweden. In fact, he did the studies for us. But he was blinded. He didn't know. When we unblinded the results for him, and we had a Zoom meeting with him, he thought it was remarkable. He hadn't seen any therapy have this effect on those spinal fluid proteins like this before. So we're really excited about that. And then the PET scans of the brain were done by Eric Ryman, one of the top people in the world in PET scans. He's in Arizona. And we provided to Eric all of our raw data. Like, it's not interpreted by us. We just gave him all the raw PET scan data. He came back to us saying, Frank, there's a significant effect on drug versus placebo on this brain scan. So anyway, good news from that phase 2 trial. And now our goal is to move on to a phase 3 trial that will be much larger.
So Frank, just give people a perspective. How much did the phase 2 cost, and how much money would you have to raise to do the definitive phase 3 trial? I mean, I obviously know the answer, but
yeah. The phase 2 trial cost probably a total of close to $20 million, and so that was obtained partly from private investors in our company, and partly from NIH, and partly from ADDF. So in each phase, we always involve peer-reviewed funding, so that a third-party experts are evaluating our technology. It's not my opinion on our technology. We always have a third-party peer review. And so now the phase 3 trial, they'll have something like 1,500 people, so much larger. So now we're talking $200 million, $250 million, a lot more expensive. But there are options to fund that. It could be a pharma partnership, NIH could fund it, or some of our investors say they can raise that amount. So we'll fund it one way or the other.
So I just thought you might find it interesting to hear the journey of a scientist over 20 years to try to develop a drug out of an academic setting into a biotechnology company with really novel technology that really could be revolutionary because of its mechanism of action. This is a pill, it's not an infusion. It could be widely used, not only for Alzheimer's in multiple populations, but for other neurodegenerative diseases. And we're right at the point now, after Frank's done this for 20 years, and we've been investing with him every step along the way. So it's an example also of how venture philanthropy can really play a role in a high-risk program to keep the program going, to provide the validation from the ADDF as a nonprofit so that other investors take advantage of our due diligence and say, well, we know ADDF does that kind of due diligence, and if they vetted it, then we're more comfortable making investments in that as well. And the journey's not over, and obviously there's still a chance for failure here, but I think the most exciting time for Frank's technology in the phase 3, and so many others. We have so many other drugs now in this phase 3 stage that we don't have time to talk about unless you want to ask questions about them. It's just such an exciting time. We have the diagnostic biomarkers to use. As Frank said, the main outcome of his phase 2 trial was to show that the drug actually did something because it moved these biomarkers. So we know the pharmacology works. And to get the effect on the clinical outcomes takes a much larger study. That's basically the fact, that to see a change in cognition, you need more people. So maybe with that, Mark, I see they're there, and we're a little over time, maybe.
No.
Well, I just wanted to say one more thing, if I may. So Frank mentioned earlier on that he's also has another class of agents in development which are probably more powerful and more neuroprotective. It's called BDNF. And one of the things that's really interesting in my view about BDNF is that we know for a fact now that exercise prevents Alzheimer's disease to a certain degree. We know that. I mean, I think after 20 years of work that we've funded and others, we can say that exercise really delays the onset of Alzheimer's. And even in people with Alzheimer's who exercise, their cognition gets actually better. The question is, how does this work? And there's many proposals about various hormones that are released during exercise. It's not just a blood flow effect. But this BDNF that Frank is developing is one of the hormones that's released during exercise that goes up into the brain and is very powerful in creating new synapses. But the problem, as Frank said, is it's not understanding the biology. That's the problem. It's creating the drug. The real challenge is in the drug, is the making a drug out of something. You can discover a gene, but to make a drug out of a gene is really, really hard. It's much harder than discovering the gene. So in this case, what Frank is taking is the same strategy of taking this big molecule that can't be delivered, it can't be really manufactured very easily, practically as a commercial product, and knocking down that big molecule into a small little pill with small molecules that could be taken orally that would mimic the effects of BDNF. So we're funding that at the company also. Maybe you could tell us a little bit about that, and then we'll let Mark in and take over.
Sure. That's the area of this BDNF, brain-derived neurotrophic factor, very powerful in supporting these connections or helping us make new connections. And the naturally occurring protein goes down as we age. It really goes down with Alzheimer's disease and other neurologic diseases. So we've made several drug versions of this successfully. They're working well in many different forms of Alzheimer's and other mice, including Parkinson's mice, which we're excited about. And we're at the point now we're ready to do the testing that will move our best candidate into humans. So our goal is to replicate what we've successfully done with the first category and get this into humans as well.
And as when I presented this program to our board, one of our board members said, well, when we were talking about the fact that the exercise improves cognition, it prevents brain atrophy, as a biomarker, and he said, well, you know, exercise is great to prevent brain atrophy and it'll slow down my loss of memory, but can't you invent a little white pill that I can take because I'm a couch potato? And I said, yeah, we got a program like that. So with that, I want to turn it over to questions.
Right. We're going to take some questions in a second, and just don't worry, when this is done, we're going to be serving hot cocoa, so you'll be all warmed up. I will say also, just a perspective for a second, Frank is one of 35 clinical trials that Howard and the ADDF are funding right now. And to the very point that Howard made, and Frank, it's more than 5 years ago, is it 10 years ago now, there was a front cover article in Time magazine when we used to buy it and hold it in our hands and said, imagine the day when a single pill can cure Alzheimer's. They were talking about Frank, and you just heard where it is today. So
2016.
there you go, 2016, which is great. Okay, so yeah, go.
Dr. Fillit, give us some more information about exercise. How much? How often? Do I have time tonight to?
Yeah. Well, I think there's two ways of looking at it. The research that we funded, I'll give you an example of the kind of work that's been done. This came out of a walk I took with somebody in the Arizona desert out where we did a meeting at Canyon Ranch. And we thought about how we could look at this. So basically, the study design was very simple. We took about 50, 75-year-olds to this person's sports physiology center at the University of Indiana. And they came in 3 times a week. They did a 45-minute walk, you know, a brisk walk, but nothing fancy. They did stretching and socialization, and they went home. And the control group came in and did the stretching and the socialization, but they didn't take the walk. We did that for 6 months. At the beginning of the study, we did neuropsychological examinations, and we did brain MRIs. And then at the end of the study, it was a very inexpensive study. We repeated the brain MRI and the neuropsych. Now, the normal brain shrinks at an annual rate over the age of 50 of about 0.4% a year. And in Alzheimer's, it's about 4%, particularly the area of the brain that's responsible for memory and learning called the hippocampus. The people who didn't take that 3 times a week, 45-minute walk, their brains shrunk at 0.4% a year. And their memory scores declined according to what would be called normal aging. But the people who took the walk, their brains actually, the hippocampus got bigger, and their memory performance improved. Now that's been taken out to, if anybody knows when Lance Armstrong or these Tour de France, they're measuring their VO2 max, which is the efficiency with which their muscles use oxygen, which is a reflection of how fit they are. And we did a study using VO2 max as a continuous variable, along with BDNF in the blood and some other biomarkers to try to understand the phenomenon, to understand the biology. And it turned out that there was a direct, almost straight line correlation between VO2 max, in other words, fitness, and the rate of decline on the brain scan and the rate of decline in memory, in other words, the improvement. It kind of peaked at a certain amount, so we're not telling people to go out there and be crazy, but there is a continuum of doing more is better. And the same was true for BDNF. So what the current recommendation is from the CDC and places like that, which we've talked a lot about lately, CDC recommendations, but the CDC recommendation is about 150 minutes a week. And that's basically a brisk walk, but if you want to run, if you want to do the elliptical, it doesn't matter. I just read a recent study that the really bad thing to do is have sedentary life, because that's really bad for you. And anybody who's sat around all day feels groggy, and I think that's very real. Anybody that has depression who exercises feels better, and that's very real. That's biological. So we know in our hearts, we know from our personal experience that exercise affects our brain, and we now know from the biology that's true. But what we'd really like to know is how it works, so we could develop a pill that would mimic that and enhance everyone's lives.
I know there have been a number of studies done in terms of lifestyle factors, and they have really proven definitive. But are you then doing any studies that, in addition to the exercise, factor in eating habits, they promote the Mediterranean, diet and weight control and that kind of thing.
Right.
So I'm wondering if you're kind of, having like A, B, C, or D, and then measuring kind of if you can maximize by incorporating these other elements.
Well, one of our recent board members is a friend of mine named Mia Kivipelto from Helsinki, Finland. That's right, I got that right. And she is a world leader in this prevention research. She's done the only randomized controlled trial of prevention and showed that prevention works. So there are like 12 factors, as you mentioned, obesity, sleep, stress, diet, Mediterranean diet, exercise, don't smoke, don't drink too much alcohol, manage your diabetes, manage your hypertension, wear your hearing aids. These 12 have all been validated by something called the Lancet Commission in the UK, that if you do all those things, people who would do all those things could reduce their risk of Alzheimer's disease by 40%. That's huge. That's more than these drugs do. So Mia's study is being replicated in every major country throughout the world now to see in different populations, if you do prevention, if you do all the things that you're supposed to do, can you slow down cognitive aging and prevent Alzheimer's? And the what we call implementation science of her work is that the control group just got education and counseling. It's like you go to the doctor and the doctor gives you a piece of paper and says, here's what you should do. Go home and read this and do it. Versus a program where there's an intervention and you're kept to be compliant. And that was the randomized trial. What we're doing, if you think about how we prevent heart disease, we do all these things. What's good for your heart is good for your brain. But we add statins because we know cholesterol is a risk factor. So we can have a medical intervention that's preventative along with these lifestyle risk factors. What we're doing is funding Mia to add metformin to her lifestyle intervention program because metformin we've run clinical trials of metformin, which is the leading diabetes drug, but it's also been shown to improve the metabolism of the brain. And so there's a lot of interest in using metformin for Alzheimer's in a repurposing manner. So we're using metformin in people who are normal, cognitively normal, but they get the brain scan. So these are people that have Alzheimer's disease, but they're not symptomatic yet. And the purpose of the trial is to use lifestyle interventions with comorbidity management of diabetes and hypertension, because they're risk factors, along with an anti-aging drug, and see if we can really have a big impact on preventing cognitive decline and preventing the accumulation of the amyloid in people that don't have symptoms yet, because that's ultimately would be the goal, right? To prevent people having symptoms.
Are there populations on the planet that have more likelihood of getting Alzheimer's and populations that have less likely to get it?
Yeah, I mean, that's a good question. Are there populations or people that are higher or lower risk for Alzheimer's? Probably not intrinsically, genetically. I think it's more the different populations having different lifestyles. That would, people that as part of their living situation are getting more exercise or eating in a more healthy way, you might see lower rates of it. But I think in terms of, just genetic propensity, I think just about everybody's at risk.
Yeah, I mean, the Japanese tend to get more vascular dementia, which is the second most common cause of dementia, the vascular disease of the brain. But I think another way to look at it is African Americans are more at risk. And it doesn't look like it's necessarily related to socioeconomic status. Hispanics are more at risk, and again, it's not clear why. So there's some diversity issues here. And women are more at risk, so we don't exactly know why. And again, it's how do you do prevention studies? That's the really interesting question, because when women are more at risk at age 75, how do we find out what it was during the course of a lifetime that caused that increased risk? Well, the one thing we know about women that's different from men is women experience the menopause. And at the time of the menopause, there's dramatic decline in estrogen. Estrogen, like BDNF, like nerve growth factor, is what's called a neurotrophic hormone. In other words, it's neuroprotective. in the brain. There are estrogen receptors in the brain, in men and women actually, these trigger signals in the cell that are protective. So when, and 60 to 70% of women at the time of menopause complain of memory difficulties and emotional irritability, a whole host of psychological symptoms. For most women, that goes away. So there's a really interesting study that was done recently by Lisa Mosconi at Weill Cornell and one of our board members from the University of Arizona. They took these brain scans that I've been talking about, the amyloid brain scans that we invented, helped to invent at Penn, that's been used so many times, and looked at women at the time of menopause and said, gee, I wonder, when women get this dramatic drop of estrogen at the time of menopause, is it bad for your brain? So they did amyloid scans, the amyloid scans, and what turned out is that women who didn't take any hormone therapy, they started depositing amyloid during their menopause. You could see it on the brain scan. The women who took Premarin, there was no difference. But the women who took 17-beta estradiol, which is the naturally occurring hormone that women produce and what is lost during the menopause, the women who took 17-beta estradiol did not accumulate amyloid during their menopause period. So this tells us that there is something happening during the menopause that starts a process that ends up as dementia 20 years later and could be the risk factor for why women are great at risk. And the analogy here that you probably all understand is osteoporosis. In fact, the whole brain thing came out of the model of osteoporosis. There's a window of opportunity to prevent osteoporosis at the time of menopause by giving estrogen replacement therapy. But if you let that window pass, if a woman doesn't take the estrogen at 51 and even tries to start taking it at 56 or 60, the horse is out of the barn. The osteoporosis has already started. But if women take it at the menopause at 51, the osteoporosis can be prevented. So it's the same sort of way of looking at how we can do prevention studies and ultimately design better therapies. Yeah.
Just two quick questions. Do you support the protocol that Dr. Bredesen gives in his book, The End of Alzheimer's? That's number one. And the second part is, what is your feelings on the faux biomodulation caps, you know, the via light that people now are using and can use, they think, to target the tau?
Yeah, two good questions. Yeah, so Dale Bredesen's book, so I know Dale, we were both at UCSF at the same time, and what his book talks about is going after all the factors we could possibly go after: diet, sleep, vitamins, everything. And the reason why it's somewhat controversial is because he hasn't been able to really do a controlled trial. I mean, the claim is that people's cognition is actually improved when they're in early stages of Alzheimer's or mild cognitive impairment. So that's a big claim to make, that cognition got better. But there's been no trial. So the concepts make sense that doing all that is helpful. Most of us agree that sleep and Mediterranean diet is helpful, but there's no data. So, you know, I think people like Howard and I encourage those things anyway. I think Dale Bredesen's book is just an extreme of that. And then you asked about the stimulation caps. Yeah. So there's two areas. One's called transcranial magnetic stimulation, and then there's a more recent one, 40 hertz stimulation. I don't know which one you're asking about. I can comment on both. The transcranial magnetic stimulation is putting magnets around the head and the brain and get some kind of magnetic stimulation. And this is currently, one form of this, or many forms, is currently approved for depression treatment by the FDA. So psychiatrists use this. There's been studies for at least 10 years in Alzheimer's and no consistent results. And so that's why you don't see us jumping into it. I think more work needs to be done. There's the more recent 40 hertz stimulation that was started by a group at MIT. That's been in the news more recently. So a little bit more recent in the Alzheimer's mice, it shows some changes in brain function, which is encouraging. An early phase trial in human Alzheimer's is just near wrapping up. It's a complex mixture that they're seeing. Some aspects of inflammation are increased with the idea this might remove some amyloid. So it's not something I would do outside of a trial, because, we're seeing some increase in inflammation, maybe some removal from amyloid. You can buy these 40 hertz things on Amazon, but I recommend to my families just, wait another year or two to really see what the formal results of a trial are. Is that, well, I think that's part of your question.
Okay with just brain mapping? Just doing the brain mapping?
What about the brain mapping?
Are you okay with that?
Oh, yeah, I think just mapping the brain is fine, just to observe. There are these clinics that claim all kinds of brain imaging and brain mapping. There's one that's pretty well known, but it's not really rigorous. And so, you could ask me more about that. I have to be careful who I comment publicly, but a lot of this stuff is just marketing.
And I'll just add, in terms of what Dr. Bredesen is recommending, that several of the things he recommends are supplements, ashwagandha, for example. And we, about 15 years ago, developed a website called cognitivevitality, all one word, .org. Last year we had 1.4 million visitors. And we have 3 full-time PhD scientists at the foundation, sponsored by a private donor, who are writing reports on these things. They're technical reports on the website, and then there are reports that are friendly for laypersons. So ashwagandha and bacopa and these other supplements that are commonly used. I mean, I get patients coming in with a suitcase full of, they're on 30 different things, and we don't know what's in these things. We don't know if it's poisons in these. We don't know how much good stuff is in them. There's not really proof. So if you're really interested in seeing what he's recommending and what our scientists are rating these on in terms of safety and efficacy, this website is cognitivevitality.org. Thank you.
Howard, excuse me. So what are you going to tell women my age who, when we were going through menopause, we were told, don't take estrogen replacement therapy, that it was not good for you?
Yeah, there is controversy about taking estrogen, starting estrogen after the menopause. I mean, I know people who have done it, and there was one study, the Women's Health Initiative, that enrolled over 100,000 women and studied the impact of estrogen replacement therapy in postmenopausal women. So the average age of the women in that trial was 65. And although the trial was criticized, what it showed, contrary to what we thought, which would be that taking estrogen at 65 would be protective and all that taking estrogen at 65 actually increased the risk of stroke and dementia, and even heart disease, rather than decreasing the risk. So unless there are other symptoms of menopause, like hot flashes or vaginal dryness or things like that, sleep disorders that are related to that, personally, I don't think most gynecologists these days would be recommending estrogen replacement for women that are many years post-menopause.
Doctor, have you now, or are you planning to do any experiments with CBD or cannabis?
So CBD is really interesting, and the pharmaceutical industry has tried. So in the brain, in the normal brain, there's 2 types of cannabinoid receptors. There's a CB1 and a CB2. And the CB1 is the psychotropic receptor. So you take THC and it goes to CB1 and gives you all the highs and everything. CB2 is actually an inflammatory receptor. And we know that cannabinoids, they're being sold as anti-inflammatories. Well, in the brain, there's a CB2 receptor that is neuroprotective, actually, when it's triggered by THC and other cannabinoids. What we're doing is we're funding the Case Western Reserve. We spun out a biotech company there called Neurotherapia, and they have small molecules that don't have the psychotropic effects of CB1 activation. They only have, the small molecules have been engineered and designed through medicinal chemistry to have the CB2 activity. And that program is now in phase 1, and hopefully it will soon raise enough money to go into phase 2 and see if that CB2 receptor drug can be used to treat Alzheimer's disease. Yeah, you need a microphone.
Oh, I've got it over here.
I can repeat the question.
You can go first, and I'll go after you.
Thank you.
One cannot watch television for 20 minutes without finding an ad for some brain-enhancing supplement or drug. You mentioned something, I think, about an anti-aging drug. Is there any such thing?
Yeah.
And is any of these things that are totally on television all the time, are any of them kosher?
I have a shelf in my office, in my bookshelf, of like about 50 different supplements that, are being sold in Duane Reade and CVS, and I always look at what it is. And it's often like things like phosphatidylserine that was proven not to do anything like 25 years ago, and things like that. I mean, the most common one you see is Prevagen, right? But what I'd encourage you to do is, as the commercial comes on, in very light white lettering, it's so small you can't see it. And it says what the the FDA requires them to do honest marketing. And it says what the drug actually did in clinical trials. And if you look at the data, there's like no data, basically. So they're allowed to sell supplements as long as they don't make claims. That's the thing about the supplement industry. It's a $10 billion industry because these supplement makers are allowed to sell supplements as long as they're safe and they don't make any claims. And basically Prevagen, they can have testimonials, but you'll see that they're not really making any disease claims. And if you look at the actual, you got to catch it because it's only on for a second. It's almost not readable. You'll see that it doesn't do much. Well, the metformin, there are several. We're testing some other drugs, some senolytics drugs that are called senolytics at Wake Forest. But the metformin is one of the more interesting ones these days, I would say. That's available. It's a diabetes drug, but it can be prescribed off-label, and it's basically safe. I do prescribe off-label, and for people that I feel it would be safe for, I'll prescribe a low dose and try to explain the benefits and possible harms, particularly people that have mild memory problems and they want to do something. And I feel that I'm willing to prescribe them something based on what I think is good science, even though it's not definitive. Okay, yeah.
Thank you both so much for this really informative and just enlightening conversation tonight. I'm really curious about the digital biomarkers and digital therapeutics that are coming out and what areas you see as most promising for Alzheimer's and dementia research moving forward.
I think I can, yeah, well as many of you may know, we have now almost $100 million initiative with Bill Gates, with Gates Ventures, with Jeff Bezos, with Mackenzie Scott, with David Dolby, with the Schwab Foundation, with the association. Anyway, I won't go through all of them. And the whole purpose is to develop digital, one of the main purposes is to develop better diagnostics and particularly digital technology. So let me give you one vignette of how I see digital technology in the future. One of the earliest signs that someone can pick up, a spouse can pick up, or a friend, or even during a speech, is a change in language. People have word-finding difficulties, their grammar sort of changes, their intonation changes over the course, the speech gets a little flatter. And you kind of, lay people, when you hear that, you kind of sense like it's not quite right. Well, it turns out that with all this new speech technology, you can pick that up and analyze it with artificial intelligence. And so it means that we can diagnose Alzheimer's disease way earlier using speech and language technology than we can, when we don't have to wait for someone to come in where the spouse says he keeps asking me the same thing 10 times in an hour. And so how do you distribute that technology? Is it going to be on a cell phone? Is it going to be on your computer? Will we be monitoring people as they use the computer? Will it be a passive collection of data? In other words, people will be connected through their Apple Watch and somebody will be capturing the data over time? Or will it be active, like you'll sign in once a month and and we'll get some speech patterns from you? The other thing in terms of that's early diagnosis is how we'll use that technology in clinical trials, because it's very sensitive and it's an important clinical outcome, knowing how people's speech is changing. So it could be an additive outcome measure in clinical trials as well to see if a drug is working. And we're very excited about that, and there's many other types of digital technologies. Like right now, most doctors are using paper and pencil tests, and we're converting a lot of those paper and pencil tests into much more sensitive digital tools that can be done on an iPad, and will give doctors much more objective measurements of cognitive function in the office at a very low cost.
It's good. First of all, can we have some applause for our two wonderful doctors? Just brilliant presentation. You know, the last question was a perfect one to end on because at ADDF, 3 things, and you heard them all tonight: preventions, biomarkers, and we heard why that's important, and treatments, like you heard about tonight. 3 final words or thoughts here. One, you all can find at your seat place, save the date for our Hope on the Horizon dinner taking place on March 3rd at the Beach Club. I will tell you, Howard mentioned another doctor, Dr. Mia Kivipelto, the leading doctor of prevention in the world, will be with us that evening, so worth coming out and seeing. Second, after that, on April 26th, we'll have our final in this year's series, lecture series, and we'll feature another one of our clinical trials, Dr. Jeffrey Cummings from the University of Nevada. And again, there's information at the table as you leave on that. And last but not least, for all of you who still have questions, the 2 docs aren't just going anywhere just yet. The bar is open in the back. I don't know if there's hot chocolate there or not, but there's liquor. Okay. Thank you for being with us tonight.